Happy Tuesday, biologists. I wanted to make a video going over this document because this week is crazy pants and they keep stealing you guys from me. So we're going to go over how to do the student data sheet. So that means we'll be able to do the paternity test on Thursday to prepare for our lab that we're going to do on Friday. I'm really excited about that. So remember, we're talking about gel electrophoresis. And we're trying to see how is genetic technology used to analyze DNA changes to determine genetic expression and relationships. How fun is that? Okay, so part A was talking about restriction enzymes. And restriction enzymes are really important in splicing DNA and splitting them up. And that helps us because DNA strands are very long. And they're much easier to read if we're able to see the different strands, how long those strands are, and all that good stuff. So restriction enzymes break the phosphate bonds that hold the DNA nucleotides together. And it generates a 5' phosphate and a 3' hydroxyl group wherever it's at the point of cleavage. Remember, cleavage is the point where it is splitting up, not the other kind. So it gave us some examples of different restriction enzymes and then their recognition site. So what this means is this is the point where this restriction enzyme recognizes the DNA strand and the nucleotides, and that's where it knows where to cut. So that's what those little arrows mean. So on your handout, we have the instructions here. So I know it's kind of cut off weird, but it's fine. We're gonna keep it because I'm gonna commit to it. So the restriction enzyme HINC2 recognizes the base sequence GTC-GAC. It'll cut the DNA strand between the C and the G. So let's go to your student data sheet. Here. Everything's kind of wonky. It's fine. Actually, not here. Here. Okay, so I already forgot the DNA strand. GTC-GAC. Okay, so GTC-GAC, we're going to highlight that. You guys are going to highlight with a real highlighter. And you're also going to make the 3' strand, so the non-template strand. So remember, in DNA, T bonds with A bonds with T, A bonds with T, G bonds with C, G. Wow, it took 100 years, but it's fine. So we're also going to highlight the GTC-GAC nucleotides on the opposite strand. So it should be right down the middle. And it said it splices, it cleaves these strands in between the C and the G. So I'm going to use the little draw tool. And I'm going to cleave it in between the C and the G and between the C and the G on both strands. So this is what it should look like. Okay. Number 2, ask us to, we've already done that. The restriction enzyme EcoR1 recognizes the base sequence GAATTC and cuts between the guanine and the adenine. So this is going to be a little bit different. GAATTC. Let's go back down. GAATCG. GATTC, here it is. So we're gonna highlight here. Cute. We're gonna make the other strand. So
okay, you guys ready? C, I'm gonna speed this up, it's fine. G bonds with A bonds with T, A bonds with C, whoops, that's fine. G bonds with C, A T, A T. This takes a long time on the device, it's fine. C, Y, Y, C, and G. You guys should absolutely know how to do this. There's the link to the quiz. Okay? So
we're going to then highlight the opposite. So it was, double check, make sure I'm highlighting the right one. Number 2, GAATTC. Okay. GAA-TTC, GAA-TTC. So it should be the opposite on the bottom strand. But this one is a little different, like I said, and it is cut between the guanine and the adenine. So this isn't going to be a cutesy little middle slice this time. Nay, nay. You're going to slice it between the G and the A and the G and the A. And whenever the slices aren't like number one, just right in the middle, you'll just connect these slices. So much fun. Okay, so now we want to count how many fragments of DNA are for the eco-RI, which is the second one, and how many base pairs. So now you need to know the difference between fragments and base pairs. So the fragment is the actual line of DNA. So this would be considered a fragment. This would be considered a fragment. And then of course the GT, C, and A are considered the base pairs. So for this one, it's cut in half. So we still have 2 fragments of DNA. But the number of base pairs is different because the slice isn't exactly in the middle. So we'll count the base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9. Nice. Okay. Let's see if it's the same. We're going to start at the end of the slice. 1, 2, 3, 4, 5, 6, 7. So for the first fragment, we have 9 base pairs, and the second, we have 7. Alrighty. Okay, so now we've done number 1 and number 2. Let's go back to our instructions. Those of you with physical copies don't have to do as much scrolling. Okay, number 3 says answer questions on data sheet. So we scrolled all up here for nothing. And then number 4, oh, we already did 3. Number 4, highlight the eco-RI recognition sites.
Teachers and students, please pardon the interruption. At this time, all teachers, please send a count of the number of students who are present in your class to the main office.
Like you guys are still
Send
here.
a count of the number of students who are present in your class on bodies right now to the office. Thank you.
Okay. I'll do that in a moment. Highlight the eco-RI recognition sites on both sequences. So remember the eco-RI is the same as number 2, GAA-TTC. We know that because it says eco-RI or eco-R1. Draw cleavage lines. The 2 strands are similar, but DNA strand B is a mutant. Woo! And we're going to use the fragments in part B. And then we just answer 5 through 7. Okay. So we're looking for GAA-TTC. No, GAA-TTC. Okay. Ooh, not this one.
4. You got 4?
Yeah.
All right.
Thank you. All right. And then we're going to highlight the one on the corresponding strand. Okay, let's see if we have any more. Oh, we have got another one. GAA-TTC. GAA-TT. Hello. Oh, sure. There we go. Okay, so we've got 2 on this one. How fun. Let's go ahead and do it on B. So we're looking for GAA. Nope. GAA-TT. It looks like for this one, we can see the mutation. So we have GAA-GTC. So the T, the thymine, changed to a guanine. Okay, so strand A, strand B. All we're doing is looking for these recognition sites. Easy peasy. So number of fragments in strand A, we're slicing. Let's draw those slices. That'll help us answer this one. So in between the G and the A, very similar to number 2. G and the A, G and the A, G and the A, G and the A. So let's count our number of fragments. So we have 1 fragment here. We have 2 and 3 for strand A. How cool, because we've got 2 slicing points. So if we cut a hole 2 times, we get 3 fragments. And then this one, we only have 1. So if we're slicing something 1 time, we end up with 2 fragments. number of base pairs in each strand for strand A. So let's count those. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay, and then we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So remember, we're counting at the end of that slice. All right, and then we have 1, 2, 3, 4, 5, 6, 7, 8. That looks a little cuter. Okay, for strand B, we should only have 2 numbers because we have 2 fragments. So let's count those little nuggets. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28. And then this one is the same, so I can do a little cheating. I already know there's 8 on this side, so 28 and 8. Yay, okay.